everyone. In this lecture, we are going to discuss about homopolysaccharides. So, in the last lecture, we have seen about what are polysaccharides and their introduction, and also about the classification. So, the polysaccharides are basically two types: homopolysaccharides and heteropolysaccharides. So, among homopolysaccharides, we have already discussed about the starch and its compositions like amylose and amylopectin. So in this lecture we will see the remaining homopolysaccharides. So the second most homopolysaccharide is glycogen. So glycogen upon hydrolysis gives rise to glucose. So any kind of polysaccharides which will give glucose upon hydrolysis will be known as a glucan. So glycogen is a storage form. When it hydrolyzed, it will be converted into glucose. Then it will be utilized by the body. So it is the major reserve carbohydrate in animals. So it is called as an animal starch. So the excess amount of food in the animal body will be stored in the form of glycogen in the liver. So whenever the body needs glucose or whenever body wants to produce energy, the glycogen will be converted into glucose. Then it will be utilized by the body. So glycogen is stored in the liver, muscles of all animals. Among plants, it is found in fungi and yeast, which are devoid of chlorophyll. So the fungi and yeast also comes under plant kingdom. So these are devoid of chlorophyll, which means they don't have chlorophyll pigment. So the glycogen is also found in this fungi and yeast. So this is in the form of a white powder, dissolves in water, and the solution gives red coloration with iodine test. So when you see the structure, it is a branched polymer of glucose with both alpha 1,4 glycosidic linkage and alpha 1,6 glycosidic linkage. And the external branches of glycogen molecules contain 6 to 7 glucose units. So the branches, there are two kinds of branches which you can observe in the structure of glycogen. The external branch contains the glycogen molecules of 6 to 7 glucose units. The internal branch contains 3 glucose units. So the molecular weight of the glycogen is minimum up to this Daltons. The actual molecular weight inside the tissue may be higher than that. So it is a non-reducing sugar, again gives red color with iodine test. The red color disappears on boiling and reappears on cooling. So this is one such chemical property of glycogen. So the blood always contains 1% of glucose. When it exceeds 1%, exceeds 1%, the excess glucose, as I have mentioned, it is stored in the liver, and the liver stored the glucose in the form of glycogen. So the percentage of glucose must be 1% in the blood. If it exceeds, the excess amount of glucose is stored in the form of glycogen in the liver through the pathway known as glycogenesis. So glycogenesis. So muscle glycogen, when this 1% comes down, the percentage of the glucose when falls below 1%, then the glycogen will be converted into glucose and maintains the blood sugar level through the pathway known as glycogenolysis, in which the glycogen will be break down into glucose. Muscle glycogen utilized as the energy source during muscle contraction. So the glycogen, whatever is stored in the muscle, it is for muscular contraction. Glycogen resembles amylopectin of starch chemically but differ in molecular weight and degree of branching. So by the glycogen, what is the similarity between the structure of glycogen and amylopectin is, because these two consists of two kinds of linkages, alpha 1,4 and alpha 1,6 glycosidic linkages. So that's why glycogen resembles to amylopectin of starch chemically, but the molecular weight of the glycogen will be more than the amylopectin of starch. And also there will be difference in the degree of branching. So this is the structure of glycogen, as you can see here. So you can found two kinds of linkages, alpha 1,6 linkage and alpha 1,4 linkage. So it is somewhat similar to the amylopectin structure in case of these linkages. Let me show you the amylopectin as well. So this is the amylopectin do have two kinds of linkages, right? So similarly the glycogen does. This is the structure of glycogen. So third most homopolysaccharide is cellulose. So cellulose is a non-sugar. It is composed of again glucose units. It is not easily hydrolyzed. It is hydrolyzed to glucose by concentrated H2SO4 or concentrated NaOH. So it is not easily hydrolyzed. So for its breakdown, there must be concentrated sulfuric acid or concentrated sodium hydroxide, a strong acid or a strong base. So the main constituent of cell wall of plants. So cellulose is the main constant of the cell wall, right? The cell wall is mainly composed of cellulose, and cellulose occurs in lignin and cotton. So the molecular weight ranges from between 200,000 daltons to 2 lakh. That is, it may contain 1200 to 12,500 glucose unit per molecule. It is formed by the dehydration of water between the glycosidic OH or carbon atom 1. So which means it consists of which kind of linkage here? Obviously we know that whenever there is a formation of a glycosidic bond, there must be a loss of water molecule. So this glycosidic bond formation will be between the carbon atom 1 of one ring and carbon atom 4 of another ring. That's why the beta 1,4 glycosidic linkage is present in case of cellulose. Again, it is tough. Cellulose is tough. Cellulose is tough and white solid. It is insoluble in water. It dissolves in ammoniacal cupric hydroxide, which is also known as Senwitzer's reagent. And it does not give any color with iodine test. It is not nutritive as well because it helps in the structural compositions of cell wall. So because of its inertness towards the chemical reaction, it don't have any nutritive value, because whenever you want to break down, whenever you want to test the cellulose, it will not react easily with the chemicals or with other substances. So due to this inertness, it don't have any nutritive value, and it serves the purpose of structural composition in case of plants. It is not digested by man, of course. Ruminants like cattle, sheep, goat, camels, giraffe, and certain wood-eating insects are able to digest cellulose due to the presence of the digestive enzymes in their digestive tract. So here the man won't be able to digest the cellulose. So the ruminants have the digestive organs in such way that they could be able to digest the cellulose, and also the digestive tract is rich in enzymes like cellulases which break down the cellulose. It is used in the manufacture of paper and mercerized cotton, cellulose acetate, etc. So these are some applications of cellulose. So this is the structure of a cellulose basically. So this is the cellulose structure. So these two are cellulose structures in two different ways. This is the simple basic structure, which means you can easily remember this structure. So this consists of beta 1,4 linkage here. The glycosidic bond is actually between 1 and 4 carbon atoms between the sugar units. So here these are the glucose units which are monosaccharides. Between there are glycosidic bonds which are beta 1,4. So forms a cellulose structure. The next one is chitin. Chitin is also important polysaccharide of invertebrates. So invertebrates, it is formed by the polymerization of N-acetyl-glucosamine by beta 1,4 linkage. So there is a compound known as N-acetyl-glucosamine. So the N-acetyl-glucosamine, between the units there will be the formation of beta 1,4 glycosidic linkages, leads to the formation of chitin. The chitin is related to cellulose. So the cell wall, the fungi do consists of cell wall, right? The cell wall of fungi is composed of chitin. And you see the exoskeleton of cockroach, right? The exoskeleton of cockroach is also made up of chitin. So as it contributes for the formation of cell wall, it is related to the cellulose. The alcoholic OH group on carbon atom 2 of beta D glucose units is replaced by an acetylamino group. So the glycosidic bond, the alcoholic OH on carbon atom 2 of glucose unit will be replaced by the compound N-acetylamino group. It is found in the exoskeleton of insects, as I have mentioned, and crustaceans, and in the cell wall of fungi. So in case of crabs, exoskeleton also you can observe the chitin. Chitin on hydrolysis with mineral acids gives glucosamine and acetic acid. And chitin decomposed to N-acetyl-glucosamine by chitinase present in the gastric juice of snails or from bacteria. So as N-acetyl-glucosamine linkages together form a chitin, so obviously the chitin upon the action of an enzyme known as chitinase will gives N-acetyl-glucosamine as product. So this chitinase enzyme present in the gastric juice of snails or from bacteria. So you can see the structure. This is the structure of a chitin. So the chitin consists of N-acetyl glucosamine rings. So these are the N-acetyl glucosamine rings individually. So when you link up these two rings by the glycosidic bond like this at 1 and 4, so this is the N-acetyl amino group. N-acetyl amino group. So this is the structure of chitin. So this is the N-acetyl group. This is the in detail structure, and this is the basic simple structure of the chitin. So when you want to learn in detail structure, so this is the glucosamine group and this is the N-acetyl group. This is the N-acetyl group, so there will be glycosidic linkages. So as the OH group will be replaced, right? So the glucosamine and N-acetyl group linkages with the glucose unit form a chitin structure. So n represents the number. As it is a polysaccharide, you can draw the n number of these layers, these structures. So the last one is inulin. Inulin is homopolysaccharide. It is the major reserve carbohydrate of many plants. So actually it is the storage form polysaccharides in many plants. So this is again non-sugar with 30 to 35 fructose units per molecule with a molecular weight about 5000 daltons. The 6 carbon atoms are situated in a 5-membered furanose ring. So as it is a 5-membered ring, it is also known as furanose ring. It is formed by the eliminating water molecule again due to the formation of glycosidic bond between carbon atom 2 and carbon atom 1, which is beta 1,4 glycosidic linkage. So upon hydrolysis, this inulin gives a small amount of glucose in addition to fructose molecule. As this inulin is composed of fructose units, upon hydrolysis it will give fructose and small amount of glucose. So it is a white powder insoluble in cold water but soluble in warm water to form a colloidal solution, not a homogeneous mixture. So it does not give any color with iodine. It is used in physiological investigation for determining the rate of glomerular filtration. So it is one physiological application of fructose. So this is the structure of inulin, having a furanose structure in which the six carbons uh, are inserted in a five-membered furanose ring. So n represents again the number. If you want, you can continue the structure of the inulin, which will give a branched chain. So this is the fructose ring. So fructose rings consists of keto group here, and this is one glucose units. These are the fructose units. So there will be n number of molecules of fructose and small amount of glucose. So these are the homopolysaccharides, which is one type of classification of polysaccharides. So in the next lecture, we will see heteropolysaccharides. I hope you understand this lecture. Thank you for watching.